This study was about to understand the differences of at ancestry level, and we right now are focusing on colorectal cancer in Latino populations. We focus on first generate clinical genomic data from our Hispanic community around our catchment area here in LA. So what we try to understand here is if there's any difference in mutations in colorectal cancer from Latinos that basically has um, a difference in the tumor burden mutation. At this point, we want to see if young people with colorectal cancer have different mutations than people that have more than 50 years old or late onset colorectal cancer. So one of our main questions is to understand why Hispanic Latino in LA are experiencing high levels, high prevalence, and high incidence of colorectal cancer. And this is our main question. We designed the study based on, first of all, go to the community, obtain tissue from different surgeries, to extract parts of the tumor, and then at some point using clinical and genomic tools to analyze the genomic part and then integrate with the clinical data in order to better characterize the tumors. So the most important part here is that we characterize colorectal cancer tumors from early onset colorectal cancer in Hispanic Latinos. And once we obtain this information, we identify that there is a difference between the early onset and the late onset at the mutation level. We don't know if this could be a different biology, but we just found a difference. Now, beyond that, we perform ancestry analysis in which each of our patients got an analysis of their genome. And based on this genome, basically, we identify 5 superpopulations. So Americans, Africans, East Asians, South Asians, and Caucasians. And based on these 5 superpopulations, we identify that the individuals that have higher percentage of genes pertaining to one specific superpopulation that is based on the Americans have a higher incidence in colorectal cancer, specifically early onset colorectal cancer. And I think this is one of the most important discoveries we have done until today in my lab. Yes, we identify also that specific genes like APC, TP53, KRAS, and even MYC had different frequencies. We compare to other studies in worldwide, try to understand if the Hispanic Latino population in LA had a different tumor mutation burden. And of course, we identify differences, statistical significant differences, and that's really helped us to understand a little bit more about the biology. Now, another important point that we did is about we perform a spatial transcriptomic analysis. We try to understand at the very tissue level how different cells inside the tissue of the tumors are communicating. And at some point, there was a very interesting results where we identify that the stroma, the cancer cells, and the immune cells have a very particular relationship that we are describing at this point and understanding more. And we want to expand these results and increase our sample size and show more relevant and better discoveries according with this very top age technology that is basically spatial transcriptomics in colorectal cancer. And I will say that for first time in this lab, And probably one of the first studies in spatial transcriptomic colorectal cancer in Hispanic Latinos, where we are for first time trying to understand this interaction of the immune cells, the stroma cells, and the cancer cells in the tumor. And when we study for first time this, we describe a very interesting biology never described before. And this interaction will help us to target probably better therapies in order to attack these cancer cells that probably are having a different behavior than other tumors in this specific population. And probably that's the cause of the higher incidence and prevalence of colorectal cancer in young population, in young Hispanic Latinos in the LA area. Well, I probably will say that when I talk about the AI conversational tool, one of the things that I want to highlight is we generate for first time an artificial intelligence conversational tool that is a technology that help us to have a conversation with a machine that in the background have terabytes of data, tons of files and tons of packages and tons of analysis already embedded that can help us to increase our chances for discovery and to understand better the biology and to identify better treatments and potential better uh, uh, therapies for cancer tumors.